This is Cheloniacast, episode 33. Today we are joined by Dr. Natalia Gallego-García, who's currently a postdoctoral researcher in Dr. Brad Schaefer's lab at UCLA. Dr. Gallego-García has works predominantly in the area of conservation genetics with turtles and tortoises, and is a collaborator with WCS in South America. We're really excited to talk to her today. You'll also notice that Jack and Jason, some of the other guys that are usually on here, are absent today. I'm starting this one off solo, but they're currently doing turtle work as we speak, so they're excused. But thank you for joining us today, Dr. Gallego-García. And we're happy to have you.
Yeah, thank you for having me.
All right, so I'm gonna we kind of like to start off with one question, and just to see kind of how the responses vary is interesting. But what first got you interested in turtles and tortoises? Where did this path sort of start off?
I think I'm kind of a weird person in terms of how I started working with turtles. Like, oftentimes people will tell you that I don't know, that they live by a pond and they saw them and that they have a farm, or that their parents used to take them to the field and then they just fell in love with these creatures. But with me, it's like the complete opposite. Like, I grew up in a big city. I've never, I wasn't in contact with nature or with animals or anything like that. I didn't know anything about wildlife in general, and I was actually kind of like a normal big city person, kind of afraid of wildlife and all that stuff, because I was never like in contact. And I think it was kind of my interest in research and kind of getting rid of that fear that I had, what pushed me to study biology. And in Colombia, when you are an undergrad, you have to present, we actually call it a thesis. And it's like a big project. From my perspective, it's kind of a master's thesis that you have to present in order to graduate. And I was kind of looking for projects, and funds, of course, uh, on whatever I could potentially find. It was kind of more a fund related thing. And randomly, I got an opportunity to work with turtles in northern Colombia. And I thought, like, oh, okay, cool. Like, I know nothing about them, but it's funded and it looks like fun. So let's check out and see what comes out from this. So I got a list of I think it was three species that from northern Colombia. And I started doing like a literature research because, as I said, I didn't know anything about them. And what I found was that there was no information whatsoever about this species. So that's like the first thing that I found like interesting. Is like, oh, okay, there's nothing. So whatever comes out from these is going to be important for the species. The second thing that I found out was that they were extremely endangered. And the third one, which was kind of the thing that kind of gave me so I had to choose between these three species, and I chose one of the endemics. So I was like, okay, it's endemic, it's endangered, and no one knows anything about this species. And then that's when I chose to work with the Magdalena River Turtle, and that's that's when I started. But as I said, it was kind of a random thing that pushed me to get in contact with these animals and to learn more about them. So I traveled to northern Colombia. I first contacted my first mentor, which Professor Olga Castaño, which is like the back then one of the very few people that worked with turtles and tortoises in Colombia. And I told her that I was traveling to Northern Colombia, that I had this opportunity to work with this species. And she was at the beginning kind of reluctant to help me out. But at the end, she got engaged in this project. And actually, she got so engaged in the project that she went to the field with me. And we spent a lot of time working there. So when I started working there, I moved to Northern Colombia, and I just fell in love with with the critters and with the communities along the river, the Sinu River, and I kind of that's how I started to also go into the line of conservation when I see all these pictures. But just to kind of answer your question, it was absolutely random. I mean, like life just pushed me into that direction, but before that, that was not part of my life plans.
That's really interesting that sort of happened. Definitely seems to be the case with a lot of people. Is just pursuing something in the realm of. I assume you were pursuing something related to ecology or biology for school.
Exactly. Yeah,
Okay,
but then when you get to learn more about the species, then you realize like, oh, this species is in a really difficult situation, like we have to do something, but we can't do anything because we don't have any information. So how do we start with this problem? And that's kind of how it all begins. So okay, let's find out these, and that leads to a management decision, and then that management decision opens a big fan of other questions. So okay, then let's find these, and then let's find out that, and then you are um, dragged into the conservation and ecological research when you face a situation like that one.
so the work you mentioned, the Magdalena River turtles, and you've done some work with those, particularly one of the I think it was a 2016 paper you wrote, looking at the sort of differences in the temperature thresholds for the TSD in those species. Was that the first project you'd done with those? Is that the one where you went to that, or what were you working on prior to that?
No, no, the first projects that I worked on were more finding out life history information on that species. So, as I told you, nothing was known back then. So, the first one that I did was kind of, first of all, like exploring the distribution range within the Sinu River. I don't know how familiar you are with that species, but it has a complicated situation in Colombia. So, as I mentioned, it's endemic and it only occurs in those two rivers, the Magdalena River and the Sinu River. So, it's kind of odd for me to refer to that species as the Magdalena River turtle, but I'm actually working in the Sinu River. So, it's kind of confusing. So, specifically in the Sinu River, um, there's a dam that was built around 1995. I think it began operations like in 1995 or 99, something like that. And it was built in the head of the river. And since it was built, the whole flow of the river has been regulated, and the water level depends, of course, in the amount of water that the dam releases. And before the construction of the dam, the water level was low during the dry season and was high during the rainy season. And during the dry season, all these sand beaches were exposed, and the turtles um, used to go to those beaches to lay their eggs. But when the hydroelectric dam began operations, that dynamic went crazy. And during the dry
Right.
season, oftentimes they were releasing a lot of water and they used to flood all the nesting beaches. So there, and that river specifically is not connected to any other river. So it's kind of an isolated drainage. So you have, it's kind of a swimming pool where you have like a wall in one end and then the Caribbean Sea at the other end, and no connections in between. So the population is completely trapped in that river. So that means that the population dynamics and population growth depends completely on the amount of on recruitment of the amount of new individuals that are born in the river, because you cannot have migrants from anywhere, because as I said, it's not connected. So if you break that dynamic by flooding all nests and no one it's hatching, so there's no new individuals into the population and there's no migrants coming from other sources, eventually the population will crash, right? Because you don't
Right.
have
Yeah.
any new individuals into the population. So that's the situation currently in the Sinu River. And because the dam is already there, and we have, I mean, we have had a lot of improvement in terms of getting them to regulate the production of energy that somehow matches that natural dynamic, where like try not to release water during the dry season, but it's still really hard to get to that point. So the only alternative that we had was to literally just to pick up as many nests we could potentially find along the river and take them into the incubator, hatch them artificially, and then release back the hatchlings to compensate for that loss. So when we began that activity or that management strategy, one of the things that we needed to know was, how do we incubate these eggs? You know, what's the temperature? We call it the pivotal temperature. So you know, like with temperature dependent sex determination, depending on the temperature, you will produce females or you will produce males. And we didn't have that information specifically for the Sinu River. There was a study done for the Magdalena River population, but the question back then was like, is it the same? I mean, is the findings, are the findings in the Magdalena River the same as the Sinu? So can we just like take that information and apply it to our population, or is it different? And because no one I mean, back then there were no like published studies comparing different populations, we didn't know. But we needed to be sure about the temperature, that the incubation temperature of our population. And that's why we decided to replicate the Magdalena River study, but for the Sinu River population. And that allow us to answer our question, which was like, is it the same or not? And then compare both populations. So at the end, what we found was that the pivotal temperature, which is the one that produces, so above that above that temperature, you produce males or females, was the same for both rivers, but the range of temperatures where you could produce both sexes at the same time was narrower for the Magdalena River and wider for the Sinu River. So you have like a wider range of temperatures where you could potentially produce both males and females. And with that information, of course, we were able to incubate these nests better. So now we know that we are producing either males or females. And for our management strategy, that is related, as I said, with this story that I just told you about the hydroelectric dam in that river.
Yeah, that's really an interesting way, and I think a lot of people would just take the data that existed for the Magdalena River and then just apply that. But it's interesting that you and your crew had the foresight to say, okay, we should probably analyze this separately because it's a different system, especially for a large
Exactly.
species like that that's isolated and presumably for a long time that you'd have these differences. Why do you think that the transitional temperature range for the Sinu River was broader? What's sort of the reasoning behind that?
Um, well, there's like several hypotheses around that. I mean, like it could potentially be biologically different in terms of um, it's kind of adapting to its own environment, and maybe the Magdalena River has like some sort of difference in terms of temperature, and that's why it just drifted into that direction, and then the other one kind of drifted into another direction. um, and then the other potentially issue is that which is more statistical, it's not biological, is that like in order to fit a regression model to actually find out that better, then you need a lot of observations. And in this specific type of analysis, an observation means an incubator set at a particular temperature and replicates. And with that, you need to be sure that what you think is a male and what you think is a female is a female. And as you might already know, there's no way to determine the sex on juveniles. They all look the same, so you cannot check them morphologically. You have to check the gonads, and sometimes that's not even enough, and sometimes you need to dissect and do histological um, um, slides and actually check if you have like testes or ovaries, and that means that setting up an experimental design with a lot of observation points require a lot of nests and a lot of hatchlings that you have to dissect, and that's not that nice. So you kind of have to do it in a sense that you kind of have enough information, but as I said, like it could potentially be just related with the fact that there's kind of missing data throughout the regression that we set. So that question also remains there. But as I said, like testing it is requires a lot of thing, and for what we were wondering, which was should we use the Magdalena River temperature for our specific needs or not, then I that question was answered. Now, whatever it's like the follow-up of that kind of analysis and all the questions that you're asking me, that requires as I said, like another set of or another experimental design to actually answer that question, or at least rule out that's something biologically meaningful and not a statistical issue.
Right, that makes sense. That you were more interested in seeing if there was a difference, not necessarily quantifying why that difference was there. I've
Exactly.
worked with some like GLMs before and that sort of thing, and you kind of wonder. It's easy to pick up differences, but it's harder to actually pinpoint why those differences are occurring. And like you said, to see if it's just a statistical artifact or what's actually happening to cause that measurement is a whole. It's a whole separate section of research where it's like, uh, I feel like a lot of people don't necessarily know the struggle that aren't doing that work. So that's interesting that you mentioned that.
Exactly. And this specific one is kind of not my field of expertise. Like I'm not the person like studying temperature dependent sex determination. That was kind of a study that was feeling a question or a need for management decisions. And that's why I did not kind of follow up on that specific study because, as I said, it's not like my kind of line of research. But it is interesting. I mean,
Yeah, and you also accounted for heritability in there too, which I thought was
mhm,
fairly interesting because you once you've ruled out environment as causing those differences, you have to assume it's coming from something, and the most
exactly.
logical conclusion there is it's coming from the animal. So how do you account for that? And that's sort of more kind of your with the genetics aspect, right? Is kind of your area.
Yes. Uh, you just explain it. So, like the question was, is this just like a plastic trait? Is just like phenotypic trait, so to speak, or does this have some sort of genetic component that's which kind of says what I recently mentioned, which is like, oh, so that would mean that they are kind of adapting to their different environmental conditions in each of these two rivers. But as I said, that's another one. I mean, like the analysis that we did somehow showed that there was a heritability component, and that has been shown in other systems and other turtles as well. But because we had few data, the kind of the confident intervals of that estimate are wide. They're not that narrow. Um, what that tells you basically is that you need more information to kind of narrow those confident intervals and be sure that these have an important genetic component or not. So that's another question that kind of it's still to be answered for this specific um, system.
Okay, yeah, that makes sense. I guess sort of to kind of close out or round out the Magdalena River Turtle sort of section of things. It's really interesting work that you did with this. What is kind of the conservation status of this species now? I know the Turtle Survival Alliance covers this pretty frequently in their annual reports and such. But you've been sort of I mean, you haven't followed up with that work specifically, but you've been involved with the work that's going on here. How is that species faring?
Okay, so as I mentioned, there's two populations, Magdalena and Sinu. The main issue with that species is overharvesting. In Colombia, there's kind of a tradition that during Eastern or Holy Week, Lent, I don't know how you call it, um, they have like the main traditional dish during the Holy Week is are turtles. So over harvesting is huge, but is targeted to a specific time of the year. So pretty much no one hunts too many individuals outside that week. I mean, they do eat turtles, but it's not a big deal. But during that week, it's like a big deal. And unfortunately, the that Easter or Holy Week overlaps with the reproductive season of the turtles. And what that means is that the harvest is biased towards the females because it's easier to trap them when they are outside of the water laying eggs. So, although this has not been quantified, um, it's likely to think that there are more females harvested than males. And on top of that, the females are also larger. So there's, I mean, like they're also targeted because they're larger. Over harvesting is like the main threat in both rivers. And then on top of that, in the Sinu River, we have the situation that I told you um, with the hydroelectric dam. There are several dams in the Magdalena River as well, but it's an open system that has a lot of tributaries. So the impact of those dams are not as bad as in the Sinu River, where there are no tributaries, and as I said, you have a population trapped between a wall and the Caribbean Sea. So if you have the situation where you have adults being removed at a high rate, and you have no new individuals coming to no, you have no recruitment, uh, you will lose the population. I mean, there's absolutely nothing you can do about that. And with turtles, you know that whatever effect you're currently having, you won't see it like right away. You will see it, say, in 20 or 30 years, where it is going to be really difficult to recover the situation. And that's why the species was listed as critically endangered. So we have been doing, as I said, with WCS Colombia for many years, conservation actions specifically to compensate the problem with the hydroelectric dam in the Sinu River. And we also have been doing population monitoring. So we are kind of checking every day if the trends are improving or not. But the problem is that it's not easy. I mean, what I just mentioned, like population crashes, you cannot pick them up right away. It takes time to actually see the crash. So whatever we're seeing here does not necessarily mean that that the populations are improving or not. But so up to that point, we thought that the population was in bad shape. There was then a study done by Dr. Vivian Paez. She was able to establish um, growth rates for the Magdalena River. And what she showed in that study was that the population was actually declining, and declining at a fast rate. That's more or less one of the conclusions of her study. Um, but we recently published a conservation genomics study on that species, and that allow us to see other things. We estimated the effective population size of both populations, the Sinu and the Magdalena River. And the effective population size is kind of a difficult concept to explain, but I always give kind of the same example to people just to give you an idea of what it is. So one thing is the census size, which is the number of individuals that you can actually count. But it's not the same to have a population of 1,000 individuals than to have a population of 1,000 siblings. You still have 1,000. If you count them, there are 1,000, but they're siblings, meaning that they are so related that the equivalent is kind of the equivalent to have like two individuals, not 1,000, because they are all related. So the effective population size kind of accounts for those biases. So if they're too related, if the genetic diversity is too low, if there's reproduction biased, meaning you have 1,000 individuals, but there's just two females reproducing and the other ones are not reproducing.
Right,
So
right.
that means that in the next generation, you will have a lot of relatives. So if the effective population size kind of compensate all those biases and give you a number that, of course, is lower than the census. So if you can have a census of 1,000, your effective population size will be always lower because it's kind of controlling for all the things that I just mentioned. And the kind of the thresholds established to know if the effective population sizes are good or not is a magical number that's called the 5,500 rule. So, and what the 5,500 rule says is that if you have an effective population size um, of below 50, that's kind of a warning sign that your population is kind of doing bad. Is kind of getting into like fast into extinction. And the 500 is that you need at least an effective population size of 500 to maintain genetic diversity over time. So what you would like to see in a healthy population is values above 500, and you don't want values below 50. So that's kind of two kind of warning signals that you have. When we did this analysis with the Magdalena River population, so our surprising result was that the effective population size was of, if I remember that correctly, something like 700. It was above the 500
Wow.
threshold. And there are several reviews that have compared the census to the effective in several vertebrate taxa, and that they don't vary that much. And the rough estimate is that the effective population size, it's more or less 15 or 20% of the census size. As I said, it's always smaller. So if we have like a census of 700, sorry, an effective population size of 700, that means that the population, it's of about 7,000 to 8,000 or something. Each population. So if you compare those census sizes with the IUCN criteria, that gives you better hope for that species, meaning that the population is still, I mean, there are still several animals in the wild. But those sizes, what those sizes are not telling you is if there's a trend of decline or a trend in increase. So I'm just telling you, apparently there are more animals than we thought, although I don't know if those animals are disappearing really fast or if the population is actually increasing. But that gave us a little bit of hope, so to speak, in terms like, oh, maybe we are in the position where we could potentially change the trend, and we are not in the position where we are like, this is really bad and we definitely need to take more actions, meaning like managing the population outside its habitat in captivity, and try to reproduce it in captivity to and then rewild these populations. So that kind of gave us a little bit of hope. But overall, it's still an endemic. So that means like the range is extremely small species, over harvested, with hydroelectric dams issues, and population sizes that are still small. So it's still critical in my perspective.
It's interesting with the effective population size. It almost kind of gives you, I mean, you would be able to substantiate this, I guess, but from my perspective, it seems like that gives you a better estimate of kind of what's going on with the trend of the population, or how robust it will be to certain environmental disturbances, minus humans, because humans are sort of the one thing that genetic sort of things that make species more robust to disturbances, humans can just completely obliterate that by just wiping something out and taking it out faster than there can be any kind of anything that intervenes to save the population genetically or kind of keeps it from declining to a certain point. I don't know if that's making sense, but it's to the effect that gives you better data on how the population's doing, minus the impact that humans can have on the turtles themselves through consumption and.
Yes. I mean, that's right, because as I said, you could have the false perspective that your population is doing well because you count the individuals and the count is high. But as I said, if they're all related, then your population is not going to recover, because it's really hard to recover when you only have siblings or
Right.
highly related animals. Whereas if you have random animals, then it's easier to recover a population where you are at that state. And you will never see that if you don't um, do genetics on that population. So you could potentially have the false idea that you have a large population when you actually don't.
Right, right. That's interesting. And I think in that paper you also kind of bring into perspective the larger question of older reviews that are based on kind of limited data and maybe some outdated methodologies should be updated for conservation purposes. And that seems like a lot of times, I mean, Penn and I talk about this kind of thing a lot. We're interested in this with larger scale where you're trying to look for differences in taxonomy between species. A lot of the stuff that's based on just fragments of genetic data seems like it's, with genomic data available now, it seems like it's kind of pointless. But when you think about population genetics, there's even some level of, and the types of analysis you're doing are different in those two different realms. But for population genetics, there's also a need to go back and revise things, or what kind of things does do we have now, I guess, that we didn't have in the past that we can analyze with.
A lot, and that's kind of the question that we addressed specifically in that study that you just mentioned. Technology has moved so fast that over the course of just a decade, um, you don't know if the information that you had is kind of accurate or not. So with that analysis, again, it like the whole idea was based on our concerns about, um, should we redo the population genetics of the Magdalena River turtle or not? And the question was because that's one of the very few species lucky enough to have a lot of genetic studies. So there was one with mitochondrial DNA, then there was a follow up with another one with allozymes. Then there was a follow up with another one with microsatellites. So this is one of those weird situations where you have the same system analyzed with different technologies. And the answer through time with these different molecular markers was always the same, was the genetic diversity of that species is extremely low. And I just I'm kind of highlighting that word extremely, because we can talk about what's extreme and what's not. um, and the population structure was subtle, which was kind of weird since the, there's two isolated systems. So it was not surprising for me to see that there was no extraction within the Magdalena River or within the Sinu River, but kind of seeing that level of gene flow across both rivers is kind of weird because, as I said, they are isolated. So when I started to do all these conservation genomic things, so we said, should we just redo the Magdalena River population or not? I mean, it has 3 4 different studies and it costs a lot of money. Should we just spend that money in a new species,
Right.
in a one that we have never like assessed from a genetics perspective, or should we just redo that? And then we thought like maybe many people face this situation, like I have this amount of money and I have to make a decision whether to invest this money in a species that we don't know anything about it, or one that is of conservation concern but has been studied many times, but with other type of technology. And we said, well, this is kind of the best, a really good system to test our hypothesis. I mean, if we
Right.
upgrade all those studies with the current technology, are we going to see something different that maybe they missed or not? So we kind of compare the different results with the different molecular markers with genomics, and kind of explain where we find differences and where we corroborate the results that they got. So most of the times we, or most of the questions, we were like kind of corroborating things that they were able to observe. But so say, like, for instance, they said genetic diversity is low. Okay, but how low? Like low, like we definitely need to be concerned about species, or is kind of low, like endangered, endemic, species level? Is the structure really that subtle, or is it just that we didn't have the statistical power to actually see it, like population structure in these species? Are migration across both rivers that high, or again, it's just a matter of we don't have enough statistical power to actually do a more or a less biased estimate of the amount of migrants moving across both rivers? So with genomics, we kind of um, like reduce the uncertainty of many of the estimates that were done previously for that species. So, for instance, one of the things that we found was that migration across both rivers was not that huge. I mean, actually, the confidence intervals of that estimate overlap with zero, meaning like you could have one migrant or none. uh, the population structure was straightforward. So you had one river was one thing, and then the other river was another thing, as expected. Um, we also compared the genetic diversity levels with other endangered species to see if they were like really off or not. And we found that where it was low, of course, but kind of where we were expecting it to be, given that it's that it has a small range and that it is endangered. And we did this estimate that I just mentioned of it, the effective population size, which was not done before. We also tested different hypotheses of population bottlenecks or expansions to see if we were like kind of having a population increase or decrease. In that specific case, for instance, we corroborated past results. Like they all said that the population was declining. We corroborated that information, but we put a specific hypothesis of recent decline. So most of the times with past technology, you were able to test a bottleneck, but within a really wide time frame. Most of the times, like before humans.
Right.
And when you work with conservation, what you would like to know is during human time, are we seeing a bottleneck or not? I mean, for conservation, it doesn't matter if you had a bottleneck in the Pleistocene. Like I need to know if we are currently having a bottleneck or not. So we found several different things, and then we were able to corroborate other things. But as I said, with more certainty. So that's another thing. We started this conversation talking about, like, why do you think this is happening? And I said, this could potentially be something biologically meaningful, or this could be just a statistical issue because of the amount of data that we had back then. So one of the things that's good about genomics is that you can corroborate information with more certainty. So that gives you a better idea or better conservation decisions. And then we kind of said, okay, so if you specifically have that situation where you have to make a decision, should I invest money in a new species, or should I just redo this one that's already done? And of course, well, you have to bear in mind several things. So what's the investment? Because if the investment is not that big, then it doesn't matter. You will get a lot of information and you're not investing that money. But if the investment is large, then that can completely shift your decision. You also have to understand exactly what you're getting out of that investment, right? Because if I tell you, okay, this is the amount of money that you're investing, and you will only getting this little thing, yeah, well, no. But if you're investing this amount of money and then you're getting all these new things, then that will potentially change your mind. So we kind of got all those information. So how much does it cost? What things you can get potentially new, what things you won't get, and then kind of put all that information together to say, well, maybe if you are in this specific situation, you can get a lot more new information. But if you're in this specific situation, you might not get more from what you already have. So maybe the investment is not going to be that good. So we just kind of partition this in a decision tree, where depending where you are, you could potentially get more or not. And that's kind of the second part of that paper. So that was more aimed to practitioners having to decide whether or not going down the road of redoing a genetic analysis or not.
So yeah, more of a cost benefit analysis for
Exactly.
conservation. It's a
Mhm.
cool thing to do, and it's surprising in a lot of cases it isn't done more often. But the limitations of the technology in the past seem to have gotten in the way. But in the future, hopefully that will happen more. Maybe we can sort of switch directions. I don't think we can stop without talking about the Dahl's toad-headed turtle, and that's a lot of work you've done with that species, and
Yeah.
that's a fascinating one. But maybe you could just tell us a bit more about what you've done with those and your major findings and takeaways from those projects.
Yes. Yeah, that's another kind of cool system. So it back in the day again, it was considered an endangered species just because of range size. It was only known from a single locality in Western Colombia, northwestern Colombia, and because of the size of the range, it was immediately listed as critically endangered. So it was kind of that microendemism situations. I think it was kind of in 2013 or so, where Dr. Herman Forero and collaborators found a new population in the east of Colombia, and that stretched the range by, I don't know how, but it doubles it. And just because of that increase in range, the species was downlisted from critically endangered to endangered. Uh, and again, that makes you feel like, oh, okay, it's better. I mean, the range is it's wider, so that means that the species is not as endangered as we thought it was. But it was when we started to do kind of mark recapture studies, and I was kind of following those mark recapture studies with genetic analysis, that we found out a completely different situation. That species originally was a forest species. It was specifically inhabiting the tropical dry forest in Colombia. And the tropical dry forest is by far the most endangered ecosystem in Colombia. I mean, like the estimates are something like only 9% remains of the forest. And from that 9%, 4% is extremely fragmented. So it's a really bad situation in terms of that for that ecosystem specifically. And of course, the species began to fragmentate with the forest. So you have a situation where you have individuals with absolutely no forest, individuals with patches of forest, and individuals with kind of a decent amount of forest. So you have like all possibilities. So when we ran all the genetic analysis, the first thing that we noticed is that the population was fragmented, and that's sometimes you think that's kind of an obvious result, So if the forest is fragmented, then the individuals living within the forest are also fragmented. But if you think about, I don't know, a bird, for instance, you could have horrible, like fragmented forests, but if the birds are capable of kind of flying from one patch to another one, they will keep the populations together, even though the habitat is fragmented. So one thing does not necessarily mean the other thing. Like if turtles can just move across creeks, then they could potentially be keeping the populations together, even though the forest is completely fragmented. But what we found was the opposite. We found that the populations were really fragmented. And then on top of that, they are isolated, extremely isolated. There's absolutely no gene flow between these little patches. And the patches are very little, which is the other situation. And because the patches have very few individuals, the level of relatedness of those individuals is very high. And the level of inbreeding as well is extremely high. So we currently have the situation where we literally have families of turtles located in different patches. They're absolute families. And as we were discussing previously, that means that effective population sizes are very low. So even though we could potentially have, say, 100 individuals in each patch, the effective population size is below 50, that magical number that we were discussing, meaning the red flag, like no, this is a really bad situation, and we have to do something about it. Otherwise, the only possible outcome is extinction. But unfortunately, that was kind of before when the species was downlisted. So again, this is one of the situations when you think that you are in better shape, and then you see things from a genetic perspective, and you realize that you are not in better shape than what you think. And because of that result is where we decided to implement genetic actions to compensate for that situation. So we did what's known as a genetic rescue project. We are currently doing that project again in collaboration with the Turtle Survival Alliance and Wildlife Conservation Society in Colombia. And what a genetic rescue program means is that you literally move individuals across the families because they cannot move naturally. So you literally picking individuals from one patch, putting them in another patch, and hoping that they will breed, because that's another another story, right? So what I can do is just move you, but I cannot guarantee that you are going to breed with the locals. Maybe the outcome of this project is that locals avoid the introduced ones, and they will never mate. Sorry. So if that's the case, then the project failed, because what we want is for them to breed. And maybe what that means is that you will have to move those individuals into captivity and kind of force them to breed somehow, because that's what you want. But there are several unknowns here. So we are in the stage where we have moved individuals from one patch and we introduce them into another patch. They are kind of in enclosures. So that's another thing. We were not sure about how the reintroduced animals will behave in this new environment. So we were concerned about losing them. So of course they have radio telemetry equipment, so we can find them if they move around, and they are in a fenced pond currently until they kind of established into the new environment. And in that pond, we placed local individuals and introduced individuals, and we have an experimental design where we have local males with introduced females, and the other way around, local females with introduced males, because we didn't know if the behavior was different between males and females. Maybe males just breed with everyone, but maybe the females are the ones that are reluctant to breed with the locals. So there are many unknowns, but we are trying to see if this works or not, because we need to break the level of inbreeding that we are currently seeing in that species. And something that's kind of important to mention of those genetic rescue strategies is that you have to be careful if the species shows some sort of local adaptive traits. So you want to bring new genes into the population, right? So that's one of the things. I need new genes, but I don't want genes that might break an adaptive trait in this local environment. So with that specific species, what we found was that, so remember that I told you that we have patches of families in open grasslands, no forests, and then we have families in fragmented forests, and then we had families in decent amount of forest. What we found with our genomic analysis was that forest populations were completely different from open grasslands populations. So somehow the species is kind of adapting to that human-made new environment of not having forest. And you don't want to bring grassland genes into forest populations, and you don't want to bring forest genes into grasslands populations, because you are going to break that trend of becoming adapted to these new environments, even if the environments are artificial, like the grasslands. Uh, so because of that potentially adaptive situation that we were facing, we had to make a design where we will bring in individuals that are genetically different but adaptively the same. So meaning grasslands with grasslands and forest with forest, but from populations that were very apart, not only in terms of distance, but in terms of genetic distance. And so that kind of gave us the idea of the design. So who is going to be the donor population and who's going to be the recipient population, according to adaptive traits and according to genetic differences. And that's how we designed the whole plan. And as I said, we are currently in the stage where we have already moved the individuals from the donor population and we have placed them into the recipient population. And we are kind of in the part where we are assessing the establishment of the new individuals into the recipient population so far. So that's more or less the summary of that one.
Yeah, that was some cool work. I remember, I think 2017 in Charleston. That was I had heard you talk about that. That was the first one I'd gone to. I think I was like seventh grade, probably. But yeah, that was cool stuff to
Yeah.
hear about. And yeah, seeing all the follow ups and everything with the conservation work has
Mhm.
been interesting. So when in the genetics, there's kind of an interesting thing. So you observed at a sort of interpopulational level high uh, interpopulation, a pretty high heterozygosity, but then individuals level that it was a propensity of less than you would expect heterozygotes. Is that sort of a proper way to categorize that?
No.
Okay.
So the genetic diversity of the species overall, it's low as you would expect from an endangered endemic species. So it's not high. And the heterozygosity or homozygosity is somehow related with inbreeding as well. So you're expecting, like, if you're highly inbred, you should not have high levels of heterozygote genes or
Right, right.
loci. So basically, the levels of inbreeding of each little batch of or families of turtles were very high, and that's what give us the. I mean, that's why we decided to take like extreme actions to manage this specific situation. I don't know if that's exactly what you're asking.
Yeah, I think I sort of just kind of formulated in a weird way, but that essentially the idea that you could correlate the homozygotes with the idea that there were inbreeding. Is it because there's certain times when that's not, but you can kind of rule out other things that were causing that?
What's really hard to correlate is if your inbreeding levels are causing what's known as inbreeding depression. And what inbreeding depression means is that um, you are facing a situation where inbreeding is having an impact on reproduction, growth, or any life history trait that's important for population growth. So either you are not able to reproduce or you have a disease that will kill you young or something like that. So that means that the level of heterozygosity is is correlated with a life history trait that's important for the population to survive, meaning that you will definitely have a population decline if you don't have that. So it's very difficult to relate inbreeding levels to those life history traits, especially in turtles, because what that means is that you have to prove, statistically prove, that inbred individuals are, I don't know, maybe laying fewer eggs or laying more infertile eggs or not laying eggs compared to non-inbred turtles, or that they are not reproducing, I don't know, 2 times per year, but just one, or that they are not surviving as many years as the non-inbred one. So when you think about what it takes to study turtles, right? So they reproduce not that often. Sometimes it's just impossible to find clutches, because they are like in the middle of the forest litter, and then you just cannot find them, which is the case of this specific animal. um, or do you have to wait 50 years to actually see if a result, so it's very difficult with turtles to actually correlate inbreeding levels to inbreeding depression. There are many systems where you can see high levels of inbreeding and no inbreeding depression at all, but that's rare. So the most common outcome is that if you have inbreeding, that's a concern. So if you want to take kind of a conservative approach, even though you don't have the data to show or to prove that your population is facing inbreeding depression, the conservative approach is to take actions, uh, even though you don't have that information, because it's likely, very likely, that you are having that specific situation. And you cannot wait forever to have that amount of data to prove that you are experiencing inbreeding depression, or that your population is experiencing inbreeding depression.
Well, especially in turtles too. I mean, everything sort of life
Exactly.
history wise takes a long time. So it's like,
A long time, exactly.
yeah,
And by the
can
time you get that information, then the population might be gone. So, as I said, like if you are a conservation practitioner, you just have to take decisions with the best available data that you have. You might
right,
find later that it wasn't the best decision, but you have to make a decision. And if the very few studies that you have available are showing you something, then okay, that's what I knew back then, and that's what I decided back then. This is what I know now, so I'm going to change some stuff. Maybe that's what you will know in the future. Maybe you will just change everything. But that's like a normal day in for a conservation biologist. Make
right.
decisions with the best data that you have available.
Right, and it's an exciting field too, from the perspective of being in this moment in time with it, just all the technological advances and such. Well, we can start to sort of wrap up. I've got a few quick questions that we can just finish with. Like, we focused a lot on the research side of things, and on the research side, there's also the practical side. And you've done a lot of field work, which I think is, there are a lot of people in conservation genetics that do, but some of them sort of are mostly in the lab. But as I understand it, you've done a lot of in situ work. Maybe what were some more memorable experiences, or stuff that stands out from field work that you've done, or favorite place to work, that sort of thing?
Well, I mean, as potentially any field biologist will tell you, there are many stories behind each trip.
Yeah.
Obviously, I the Magdalena River turtle is kind of um, my baby, so to speak, and everything like along that species and what I've been doing with that species of course, a special place in my heart. But I've been like moving away from that species and becoming more familiar with other species, kind of this whole world has opened for me. And you just said something that's kind of true. Like, people oftentimes think that conservation genetics is being done mostly in the lab, which is true, but it doesn't make any sense if you don't understand exactly what's going on in the wild. Like, say what we were just currently talking about, the Dahl headed turtle. So I could have ended up with results like, okay, so I have like fragmentation, but that makes absolutely no sense. And like if you don't see the environment, okay, there's no trees here, there are trees here, there's grass here, there's no grass here. So you also need that part to actually make sense of the results that you're getting from the lab. So it's kind of important to kind of keep doing these in the field. And in terms of, I mean, stories, I don't know, I have so many. um Let me just think about one specifically.
Yeah, it's always the case.
Yeah.
Everyone's got tons, but yeah, it's tough to pinpoint one or two. But yeah, I mean, anything. I guess it stands out. The the I don't know. I think you've been sort of involved with the NAFTA too, and the Belize work, that
Yes.
sort of thing. I was there, I think, in the second wave, and just yeah, the amount of that was my most recent trip, and just in 9 days, the amount of stories you come out with could fill a book. So it's just just going
And in
through.
terms of genetics, the same thing. I think that's the first time where I was kind of complaining of the amount of samples that I was taking. And not, I usually complain because I don't find animals and I don't have enough samples. It's like looking and looking and looking for animals and just empty hands, right? So that's my normal complaint. When I went to Belize, they were just capturing and capturing and capturing so many individuals that I had, like, I don't know, 15 ready to be bled. And I was like, I cannot bleed that fast. And people were like, that's actually one of the things that it's, speaking of field things, these guys are really fast, like they just captured animals, they measure them super fast, they take notes super fast, they process them like in 2 seconds, and they release the animals. But finding the veins to bleed a turtle, oh, believe me, that takes a lot of time. And especially because each species is different, So some species, I don't know why it's really difficult to bleed, and then the other ones are really easy to bleed. So, and then you have, of course, it's humid, it's raining, there are mosquitoes, they have to keep moving, they need to check 1,000 traps, and you are the bottleneck, right? You are the one delaying everything because you're trying to bleed the turtle, and it's not working, and then you have to find another vein to bleed, and then you have, I don't know, 10 people just staring at you with that face, like, okay, just please do this faster. And then you're more nervous, and then you're not getting the vein again. So what I feel is that I'm always the one delaying the team, like they need to move along, and it's me always like delaying everything, and that's horrible for them. But I mean, like, they know the deal now, and I think that they have adjusted to that.
Yeah, that I mean, you're also getting the information that nobody else can, so it's sort of that there's that part of it, but, I mean, yeah, that's
Yes. Especially, like, I remember this one time in Belize where they were like, so I was like, what's that species? And I was kind of bleeding one, and then what's that? And they said, like, I don't know. I mean, it looks like a white lip, but it also looks like a Tabasco mud turtle. I don't know. I mean, like, phenotypically speaking. So they were like, you tell us. And you just take blood of this individual, and then you will let us know what it is, because it looks funny. It doesn't look like a pure white lipped. Um, so yeah, there's that as well. Like, I can come back to them and tell them, oh no, listen, what you thought it was species A is actually a hybrid between species A and species B, or something like that.
yeah, right, that's I've been in a lot of groups where there isn't a geneticist on hand, and you find something and say, "Oh, we could just take blood from this and figure it out." But yeah, it's even a few weeks ago we were moving snapping turtles at a pond where they were overstocked, and there were certain ones in there that had kind of characteristics of a bunch of different types, and it's just, oh, you wish we had someone to take samples of things. There's always that, but yeah, when there's actually a geneticist on hand, it's sort of a different you've got that added pressure to it, but that's
And
fun.
talking about snapping turtles, like I've never seen one before. The first one that I saw was in Belize, actually. They're huge, right? And the tails are like super long and they're just huge. So I was trying to draw blood from the first one that they captured, and of course I was like super subtle, and then the needle was like shallow, and then one of the guys said like, "What are you doing?" And I was like, "I'm trying to find a vein." And he was like, "It's a snapping turtle." Like, like go deep. I mean, like the tails are huge, and he was right, right? Like I mean, it's a thick tail, so the vein runs like in between, meaning that you have to go deep with the needle, or you will never find a vein. But it was a completely new species for me, so I was kind of looking for it like superficially, and it was very deep. So that's I always remember that story as well from the snapping turtles, because that was a completely new species to me, and they're huge.
Yeah, the common snapping turtles down there are the I guess the Central American snapping
Central
turtles.
American, yeah.
Yeah, those ones are interesting. These were we were moving alligator snapping turtles, so these were the biggest one we moved was 142 pounds.
Never,
So yeah,
never seen that one.
yeah,
Like,
no, yeah,
cannot imagine.
they're cool to see, and they're if you know where to look, you can find them for sure. It's a really interesting species. We were out trapping with Dr. Travis Thomas, who does work at University of Florida a few weeks ago, and right after that event where we moved him off on the pond, we went there to Florida and did that, and that was cool stuff. But I'm sure if you ever reached out to him or anything, if you're in the U.S., that he'd probably be down to show
I
you stuff. Or
would
yeah,
love to see them in the
yeah.
wild.
Yeah, I mean, I'm not as involved with that, but I can put you in touch with people, and I'm sure he just reach out to someone. Eric Muncher, he knows.
Yeah,
Yeah,
yeah. He has promised that several times, and I will collect those promises eventually.
yeah. It's. I've still never been out to find him with him. He's seen the one. I haven't seen the Western species, but I've seen the Apalachicola and the Suwannee species. So, yeah, I have to get out with him. But yeah, all right. So we can sort of wrap things up here. But we do something at the end where I keep forgetting to tell people before this, but it's turtle trivia, and we can do this however you want to do it. If you want to ask us a question or two questions about turtles, or we can ask you something, or however you want to do it. Or if you've got to go, we can just end it now. But I don't know if you want to do that, but we do it with everybody. But I'll just throw
But
it up
meaning,
there.
like, I ask you a question, a turtle related question, and see whether you know the answer or not, or
Yeah, sure. Yeah, just turtle trivia. Yeah, that the
okay. But what? Hmm.
I normally mention it in my email when I invite people to the show, but I keep forgetting. I forgot in the past four. So, but yeah, sorry to put you on the spot, but just any sort of random. It's an easy way. One of our previous guests said it's a good way for us to demonstrate our useless turtle knowledge that can't be used anywhere else. So, yeah, I don't know if
That's
you.
a really good way to put it. But so just give me like an example. Potentially, I
Okay.
won't know the answer to that, but
All right. You want to? Okay, we'll give you a question. You got a question, Ken? Ken's got to step in here. He's got a question.
Um, Name one amidid without cloacal bursting.
um, I don't know. Say what?
Diamondback terrapins. They, being in
Oh,
brackish water, they're unusual for amidids in that they actually don't have any cloacal bursting.
didn't know that.
Yeah, something. Yeah, that's a good one though. It's fairly obscure, but just
Mhm.
yeah, there's no boundaries. Any sort of turtle related questions work.
Um, okay, so, but obviously, I will just have to say something kind of South American because that's where I know
Yeah.
the most.
Go ahead.
Oh, maybe like, can you name, say, at least what, 3 or 5 tortoises that occur in South America?
Okay. Yeah. Ken, you got one of these.
I don't.
Okay. Chaco tortoise.
Yeah, that's one.
That's one. And then the sister species to that, Galapagos tortoises.
Which are several.
Which could be a bunch. I mean, well, let's see. I might be able to name all the Galapagos tortoises, contemporary ones at least. Oh, the Latin names, I guess I'll do that. Cheloniacus darwini, Cheloniacus vicina, Cheloniacus guntheri, Cheloniacus Becki, Cheloniacus porteri, Don Fastoi. All right, that works. And then red-footed tortoises, yellow-footed tortoises.
That's it.
Yeah, I think that's all of them, right?
That's all. I mean,
All
we
right.
have all the Galapagos and Chilensis and the yellow and the red-footed tortoises, and that's it.
All right. Yeah, that's a good one though. Yeah, all right, cool. All right, we can end it here. That was thanks again for coming on. It was a pleasure to talk with you and to hear more about your work and all the
No,
cool
thank
stuff
you for
you're doing.
having me. It was really
Yeah,
fun.
yeah, thank you. And hopefully, are you going to TSA the next meeting?
I hope so. Yeah, yeah. Are you coming?
Yeah, I think I should be there. I'm not 100% sure yet, but Jack Thompson, who wish he could be on today, but is out doing surveys. He'll be there. I think he's going to talk. And then I don't know about Jason or Ken, but we'll see. But yeah, thank you again,
It
Doctor.
will be awesome if we could meet in person again after all these years.
Yeah, yeah. I would. Well, were you at the last meeting?
Yes, I was.
Okay, I spoke last in August. I was there, but I guess we just didn't cross paths. But yeah, we'll have to talk at the next meeting for sure.
Okay, like now, I mean, I don't recall seeing you, but like it's been so long that
Yeah.
so
No, yeah, compared to 2017, I've gotten a little
yeah,
bit.
exactly.
All
And
right.
there's a lot of people there, like I don't know, like most of them. No, that's not true. I know most of them, but there are a few that I don't know.
Yeah.
But yeah, definitely, it will be awesome to meet you guys in Charleston again. Oh.
Yeah. All right. Cool. Well, thanks for coming on, and for everyone listening, this has been episode 33. I might have said 32, but
Wow.
yeah, we're getting up there. All right. Thanks again.